Hello everyone and welcome to my presentation. First of all, let me thank you all for tuning in. I'm Ania from the Department of Biology of Laurentian University. As you can see on the screen, our topic today is on a protective role of hydrogen sulfide from obesity-associated metabolic stress in GLP-1 regulation. I would like to thank the organizers of this conference and my supervisor who gave me the opportunity to do this wonderful project, as well as my thesis committee members for providing excellent guidance. I'm also thankful for my lab mates and my friends and family for their love and support throughout the years. Let's start with a fun fact. Did you know that your gut microbiome weighs more than your brain? I don't know about you, but this information blew my mind. The microbiome is defined as being a diverse community of microorganisms that live in our gut and plays many roles, such as vitamin production. Recently, a less diverse gut microbiome has been implicated in many health problems such as obesity. But why I'm interested in obesity? Well, because it is a disease that is now a real epidemic in Canada. In fact, according to Public Health Agency of Canada, 1 in 4 Canadians are obese, and in Greater Sudbury, the numbers are even higher at 1 in 3 people being affected. Therefore, significant research is needed to find effective novel therapies. For the treatment of morbid obesity, Canada has approved the use of Victoza since 2015. This synthetic drug mimics the function of a gut hormone called GLP-1, which stands for glucagon-like peptide 1. Now, GLP-1 helps tell you when it's time to stop eating. It is released by a certain type of gut cells called L cells. Food stimulates the release of this hormone that, in turn, signals to our brain that it's time to stop eating, meaning it is a hormone that reduces appetite and enhances insulin secretion. In our lab, we have shown that intestinal gas, hydrogen sulfide, simulates GLP-1 secretion. Hydrogen sulfide has a rotten egg odor and is produced by the gut microbes. These microbes occupy the same environment as the gut L cells. Studies have shown that obesity was not only associated with lower gut microbiome diversity, but also with a lower GLP-1 secretion. However, little is known about why this reduction occurs. In that case, could it be the components that circulate in the blood during obesity? For instance, high levels of free fatty acids such as palmitate? Now, palmitate is the most abundant saturated fatty acid in our body. We already know that palmitate contributes to inflammation and an elevation of oxidative stress by increasing reactive oxygen species, or for short, ROS. Now, normal cell activity requires a balance between the production of ROS and the capacity of antioxidants to eliminate them. When an imbalance between ROS and antioxidants occur, that results in oxidative stress, thus disturbing normal cell function. This leads directly to my next point. I hypothesize that palmitate will increase intracellular oxidative stress by increasing ROS, which will impair GLP-1 secretion, and that hydrogen sulfide, due to its antioxidant power, will decrease ROS, thus restore GLP-1 secretion. To confirm my hypothesis, I conducted experiments on NCI and glutac cell lines, which are human and mouse L cells, respectively. As for the animal model, we chose to work on mice. First, I had to induce oxidative stress, then use hydrogen sulfide to rescue the cells and restore the impaired GLP-1 secretion. In human cells, palmitate increased ROS production in a time and dose-dependent manner. The 500 micromolar of palmitate, relative to its control, stimulated ROS production by threefold at 120 minutes. In the mouse L cells, all the doses tested showed significant ROS stimulation compared to the corresponding controls, by at least one-fold at 120 minutes. We then carried on the next experiment using 1 millimolar of GYY4137 or GYY, a slow-releasing H2S donor. As you can see here, 500 micromolar of palmitate-induced ROS was significantly reduced by 25% by co-incubating cells with H2S. As for the animal study, female and male mice were randomly divided into two groups according to diets. They were either fed a CHO diet or a palmitate-rich Western diet, which has the particular smell of fast food French fries. Mice were first injected intraperitoneally, or IP, the H2S donor GYY. Blood was then collected every 2 weeks after an oral glucose tolerance test for glucose and GLP-1 measurements. As expected, Western diet elevated body weight in both sexes, and as you can see here, with the males, they are heavier and their differences are pronounced way earlier than in the females. And we look at the blood glucose, we see the same kind of difference between males and females, where you have much greater elevation in males. Now that we have established that there is a statistic difference with fasting blood glucose, it is time to move on to the actual research objective. Two groups of fasted mice on Western diet were given an IP injection of GYY or saline at 16 hours before oral glucose challenge. Glucose concentration and blood GLP-1 were measured in both sexes. As shown in the graphs, H2S improved oral glucose tolerance in Western diet males, but not in females. Figure C shows that H2S enhanced glucose-stimulated GLP-1 secretion in both sexes. Most of that response is due to the males, so I separated these data into two separate figures. Now, it is not clear whether the females have any metabolic problem, so I decided to investigate the oxidative stress state in these animals. To do so, I examined the levels of circulating peroxidated lipids in the Western diet and CHO diet mice plasma of both sexes. Levels of MDA were only elevated in the 24 weeks Western diet fed males. Now, it is possible that the females were less sensitive to the Western diet induced metabolic dysfunction, and that's due to the elevated levels of estrogen. Estrogen is known to play a beneficial role in glucose homeostasis, as low estrogen levels can lead to insulin resistance. By knowing this, we should continue to perform studies on both sexes and develop a new experimental design to better mimic and represent the actual physiological state of women. Before I finish, let me just tell you why my research is important. Well, promoting a healthy and diverse gut microbiome might be a track for future treatment of obesity, and that by using probiotics, which are good microbes found in yogurt, for example, and prebiotics, which are the food source for these microbes. In other words, the more diverse is your diet, the happier your gut microbiome will be. And on that note, I'd like to thank you once again feel free to contact me if you have any questions.